not only are we generating large amounts of data, but we're generating data of much greater complexity. So this is this protein data bank I mentioned, and this is its growth in terms of number of structures that are in it. Also, well, not every one of these, but the majority of these little blobs are actually Nobel Prizes. So the myoglobin from John Kendrew won the first Nobel Prize in structural biology, and all of these essentially, well, most of them also brought forth Nobel Prizes. These are actually drawn to scale. So you can see what's happened over time is the complexity of data up to the whole ribosome, for example, the whole protein synthesis factory in the cell. We now understand that in ways that we just never did before. And so the complexity of this data and what that means in interpretation and use, which I'm not really going to get into, but except to say we are in an era that's quite different than it was before. I've just had a discussion with the Chancellor last week about actually establishing a data science department here, or some entity that's dedicated to data science. And people would say, well, why do we need that? Don't people just do that in their labs? Well, I think we need it. You could have said the same about computer science 30 years ago. Computer science became a very vibrant and active discipline. And I think what you're going to see is data science become the same kind of thing, because it's actually critical to the future of many industries, including the drug discovery industry. Again, my biased opinions. A number of years ago, I was actually asked by the then Chancellor to tell me what bioinformatics is in one slide. So I produced this, and it never got used because it's way too complicated and way too, there's not a picture on it or anything like that. So, but it really sort of reflects things that are going on even today. So I already mentioned this notion of huge influx of data. And what we're trying to do, of course, is turn that data into information, knowledge and discovery, including in drug discovery. And these are sort of the processes that go on to reach that. At the same time, back in, it was about 2010, we actually surpassed the number of websites. There was more websites than there were people on the planet. So, we've got this incredible increase of amount of information available. And at the same time, we're actually working at higher levels of complexity to understand what's going on, all the way to higher life forms, all the way between that and DNA sequence. And all of these projects in here, brain mapping, cardiac modeling, are things that are going on this campus that really relates to that kind of integration of data. So what you'll see coming out of all of this as I move forward is, first of all, I believe it's changing the way that we do science and the way we think about science. We've been siloed for way too long. Each of these different levels of biological complexity. What we're starting to do now is move across those levels of complexity. And it's a very exciting time. It is the best of times, at least for that. So this is just, I just love this. This is one of my, this is my dream of doing science. So Craig Venter, you probably know, has the Venter Institute here. He was one of the major proponents of metagenomics. And essentially one day he just decided, I'm going to take my yacht and I'm going to sail around the world, and periodically at all these dots I'm going to scoop up a bucket of seawater, and I'm going to sequence everything that's prokaryotic, everything that's small in that seawater. And so what he did, and then of course he comes back, so he goes on this great cruise on his own yacht, and when he comes back he's got a science paper already. It just doesn't get any better than that. In any case, so this is where he went, and he collected all this data, and so we suddenly started to accumulate very large amounts of protein sequence data that we didn't have before, because we were sequencing these complete organisms. And so there was of the order of 17 million sequences on the first pass that we'd never ever seen before, right? So there was 17 million completely new proteins that no one had ever seen before. And 99% of the DNA associated with that was from unknown organisms. We had no idea what they were. So we hadn't even begun to catalogue one tiny fraction of the organisms that were in the ocean. So the idea that there is culturable organisms there, that there are compounds that can be found, new receptors, and all the kinds of impact that it could have on drug discovery is all there. All right? But as we started looking into this, there were a number of discoveries, which I won't get into. But what it did is it started the whole metagenomics revolution. So it wasn't just getting buckets of seawater. Everybody wanted to have their own yacht, right? So they basically started scooping up and doing things. They started doing soil, right? So they were analysing all the organisms in a cubic foot of soil in different parts of the world. And perhaps most interesting, of course, was actually to look at the human gut. So the microbiome project is actually sequencing all of the organisms that exist within the human gut. Already out of that, we have some pretty good biomarkers for certain types of autoimmune diseases like Crohn's disease. So you can actually now, we're getting to the point of early detection of Crohn's by virtue of doing large-scale sequencing of the human gut microbiome. So, I think this is, exciting examples of what's to come. This just illustrates the known and unknown. So this is just one particular family of proteins. I don't want to get into a lot of detail, but you can see that the red were completely unknown. This is a phylogenetic tree, so it expresses the evolutionary distance between these things. But the only point is that higher eukaryotes weren't sequenced, but in this metagenomic data, we discovered thousands and thousands of these new PTPases. That was just one protein family. So lots of very valuable and new information. But with all of that comes problems. It's always the case. When things move very quickly, it takes a lot of time to catch up. I mean, it takes time to catch up ethically, legislatively, and in terms of technology. So as a result of all of this, for example, when you go to a database and you pull out a sequence and you look at its annotation, there are estimates to say that 30% of that annotation is wrong, because that annotation has been made by propagation. So in other words, if someone says, okay, this particular protein sequence does this, and then someone else compares it to another sequence and they say, okay, it's the same, this must do the same thing. If the first one was wrong, the second one's wrong, and you get this kind of propagation. So, we've got a lot of misinformation out there right now. That's the other point. So in any of these studies, it's very important to take heed of all of that. So that's sort of the omics revolution part of it. Let's look a little at some of the other revolutions that are going on, which I think are going to impact things. So the open science and the IT revolutions, what does that bring to the table? So who's familiar? I can ask a question, but you're not allowed to answer. Only put up your hands. Who's familiar with open access and what it means? Okay, a few people. How many people have published a paper in an open access journal? So, well, you should. So the basic idea is we're changing the publishing paradigm. We're making information accessible to absolutely everybody. And so instead of paying to read it, you're paying to write it and publish it, essentially. The models that do that, the journal models and the societal models, all still work. There's now proven business models to support this. But it changes what you can do because, for example, a typical license on an open access journal article would be what's called CC BY. It's a Creative Commons license, and these are actually declared and described accurately. And it means that anyone can take your knowledge and they can do what they like with it as long as they attribute you as the original source of that information. So the ability to propagate knowledge actually, in my opinion, increases quite significantly. And it's taken a while for it to get going, but last year about 20% of all journal publications in the biomedical sciences were open access, and that number is increasing steadily. I think in 5 years, essentially, open access will be the model. And so that creates a lot more access to information than we ever had before. I think there's some interesting software is sort of going along the same paradigm. I won't get into that. How many people are familiar with GitHub? That's a good reason. One person stretching their fingers. Okay, so I won't get into that. But I mean, other areas which I think are very intriguing are the socialization of science, and also the flattening of the hierarchy, such that anyone could make discoveries and be listened to. Right? I love this fact we're in this generation. You know, when I was growing up, I'd be sitting at the dinner table, and my father would slap me around the head and say, "You should be seen and not heard." Right? Now, of course, you know, I couldn't possibly do that with my kids. Well, they're too big now anyway, but when they were smaller. But now, we went, long time ago, we went into the generation of not being seen and not heard, but now we're in the generation of heard and not seen. I mean, I've got to tell you this, because to me it's really interesting. I got a paper into the journal that I run, and it was a fabulous paper on pandemic modeling. Right? And I was so impressed by it when I looked at it, is I arranged to meet the author, because it was a single author, and she was actually here in San Diego. We met, we talked about the paper. I was so impressed with her, I invited her. She gave a lecture here at UCSD. She went to Princeton to give a lecture on the same stuff. Really nice. So what? What's special? She was a senior at La Jolla High School. She was 15 years old. That paper was reviewed by Science. So anyone can do great stuff now. They have access to information, they have access to knowledge, and under certain circumstances, obviously she's an exception rather than the rule, but it tells you the kinds of things that are coming. And so, in my opinion, so all of this is perturbing the system.